In this section of your HE Plus Natural Sciences work, we're going to get you thinking about light, and in particular about the way light interacts with atoms and molecules. And we're going to, by doing that, get you first of all to think about the way we perceive colour and what it really means when we see something as looking red or blue or green or whatever. But equally, we're going to get you to think more about the atoms and molecules and think about what's actually happening within those when they absorb or reflect light. So by doing some simple experiments, we're going to get some insights which will hopefully get us started in heading in various directions to get you to think about some really quite challenging and hopefully interesting bits of science. Now, what you all know, of course, is that white light is really a mixture of light of different colours, and that if you pass it through something like a prism, you can separate those colours. So we can get the spectrum running through from violet through indigo and blue and green and yellow and orange and red, and that what those colours represent is increasing wavelengths of electromagnetic radiation. So what we're going to do to start with are just some simple experiments where we're going to shine white light through solutions of different colours, and we're going to use not a prism but a CD, which functions as something called a diffraction grating, and in a similar kind of way separates out the colour components, just to see what happens to the distribution of colours after the light has interacted with those different coloured solutions.
So the equipment that we're going to use for this is very basic, and the idea is that it's something you could easily set up yourself if you wanted to do these experiments for yourself at home rather than just watch me doing them here. So at the centre of it you can see there is a large cardboard box, and on the top of that box you can see that I've just created a pinhole there, and that's going to act as our sort of point light source. So to provide the light we just have a torch. This has an old-fashioned incandescent bulb so that it produces a continuous spectrum of white light. So that goes above the pinhole there. And if we then look inside the box, you can see there's the white point source. And down at the bottom of the box, there is the CD, which is going to act as our diffracting element. And you can actually see the spectrum there on the surface. But so that we can focus on that more clearly, in this experiment we're going to use a webcam. So there it is, mounted on the flap of the box at the top there and trained on the CD so that it can register what the spectrum is and display it on the laptop that's there at the front. So here you can see the range of coloured solutions that we're going to investigate. So these are just common things that I found around the house. So we have some cough medicine, couple of types of washing up liquid, and some bath foam, and we're just going to shine light through them and look at what effect this has on the spectrum. So in order to do that, what we need is a container, and here it is. It's just a simple flat bottom glass container. So we're going to place that over the pinhole so that we can shine light through it and put the various solutions in there so that the light has to pass through them on its way through to the pinhole. So first we're just adding some water. So this is going to be our control. Hopefully this should transmit all of the light. So the water's gone in there, and then we'll just place the torch on the top so that you can see it's looking white there at the top, which is good. And now we'll have a look and see what's going on inside when we look at the CD. So there is the spectrum, and you can see that we do indeed see the full range from blue at the short wavelength end through to red at the long wavelength end. It doesn't look quite like it does when you see it for yourself, because we're obviously limited by the hardware we have available for detecting and displaying it, but it gives you a pretty good idea of what we can see. So now we're going to repeat the experiment, but this time instead of just water we're going to take the red solution and add that to the dish, There we go, so you can see the red colour there, and then we can put the white torch on top of that so that we can see what happens when the white light from the torch passes through the red solution. So you can see lots of scattered red light there, but the question is, what does it look like on the CD down below? So there is the spectrum, and you can see that what the red solution has done is what perhaps you may well have expected, which is that it's cut out all of the colours from the spectrum except for the red. And so the reason we're seeing red is simply that that's the only colour of light that makes it through to the solution on the other side. So at the moment it looks like colour is really going to be rather easy to understand in terms of the way the spectrum is edited by the intervening solution. Okay, so now we're going to have a go with the green solution. So there it is, and again what you perhaps are now expecting is that what we'll see is that when we shine the light through it, the light from the blue and red ends of the spectrum should be depleted, and we'll perhaps just see the green light. So there it is in place over the pinhole. The torch goes on the top, and let's see what happens. And there we are, pretty much exactly as we predicted. We can see a nice bright green band in the middle of the spectrum there, and we can just see a tiny bit of red and blue at either side, but you can see those ends of the spectrum have been enormously depleted, and so that all seems to make sense. So it seems like maybe we're beginning to see a simple pattern here, but let's carry on and look at the next one. Okay, so this time we're going to take the blue solution. There it is. Pour that into the container. Okay, and then we'll put the torch in place. You can see, there we go, lots of blue light being scattered there. And then let's have a look what that looks like on the CD. Okay, so now we see something that maybe we didn't expect. So the solution clearly looked blue to us, but when we look at the light that's come through it, we see that it's absolutely not just the blue light that's coming through. We've got all the blue and the green and indeed the yellow. All that's really changed, and you can see it quite clearly here, is that it's taken out a big chunk of the red light. There's even a little bit of red light still getting through, but you can see that it's taken out a large proportion of the red light. So what seems to have happened is that depletion of the red, but leaving the blue and green more or less intact, is what causes us to perceive blue. So now colour vision starts to seem a bit more complicated than we might have thought. So let's have a look at the yellow one and see if we can see anything else that might help us understand what's going on. So here's our yellow solution. We'll just pour that into the glass container and put it over the pinhole and see what we get when we shine the torch through it. And there's the spectrum. So again you can see this one's not simple. What we're not seeing is just pure yellow light coming through. In fact, yellow light corresponds only to a very narrow band that you can just see right in the middle there. But in fact what's coming through the yellow solution is essentially all of the green and the red light. And pretty much all it seems to be cutting out is the blue end of the spectrum. So again we've got something more complicated to think about here, which is why seeing this whole range of wavelengths is interpreted by your eye as pure yellow light. What we're going to do now is to see if we can make use of what we've learnt by recording these spectra of light emerging from different coloured solutions, and see if we can use that knowledge to help us understand a little bit about how colour vision works. So within your eye, at the back of the eyeball, is a layer called the retina, which houses huge numbers of light-sensitive cells, which we can call photoreceptor cells. And these are connected up via nerve fibers to the optic nerve, which in turn carries the information from them to the brain. So it's these cells in the retina that we need to think about in relation to colour vision. They come essentially in 4 types. The first type are called rods. When you look directly at something, the light arrives essentially at the back of the eye rather than sort of around the fringes. And the rod cells are not actually located primarily within that region, which is sensitive to light coming from what you're looking at directly. They're more involved in, for example, enabling you to notice things happening out of the corner of your eye. So they are located more in the peripheral regions of the retina, and they're much more sensitive at low light levels than the other types of photoreceptors. So they're very important in enabling you to see in low light conditions. But we're not going to talk any more about those today, because what they are not involved in is colour vision, so they provide only monochromatic information. The remaining 3 types of cells are called cone cells, and they come in 3 different types. These are the ones which are located directly in line with the front of the eye, so that they're going to be involved in recording signals from what you're looking directly at, and these are responsible for colour vision. So the key thing is that there are 3 different types of cone cell, and what we're going to be interested in is how having 3 different types of cells enables discrimination between different colours to take place. So this is time for you to have a go. What we're asking you to do at this point is to see if you can come up with an idea as to how this might work, how the interactions of 3 different types of cells, each contributing signals to the brain, enables you to deduce what colour you're seeing. So we're just going to ask you to pause the video here for a bit and give yourself time to figure something out. Once you think you've got something, or once you think you've thought about it for long enough, then continue and see whether what you come up with bears any relationship to what's really going on. Okay, so if you've had a bit of time to think, hopefully you've come to the conclusion that the 3 different types of cone cell must be sensitive to different wavelengths, or different parts of the wavelength range that corresponds to the visible region. So in a moment we're going to show you what the sensitivities of the 3 different types of cone cell actually are. But first, it might be good for you, if you haven't done this already, to actually sketch what you think those sensitivities might be. So here we've got the axes of a graph. You can see that you've got the visible spectrum along the bottom there, and what we're suggesting you do is just for each of the 3 types of receptor, sketch in where you think they're maximally sensitive and what sort of sensitivity profile each of them has as a function of wavelength. So just for fun, have a go at sketching that, and then pause the video while you do that, so that you can then have a look and see how well it corresponds to reality. So pause now and have a go at sketching those things.
Okay, so here are the actual sensitivity curves for the 3 types of cone receptor, and as we anticipated, they each have a distinct curve corresponding to a different wavelength range. The really important thing though
is that the curves overlap. If they didn't, if you just had each cone being sensitive to its own unique wavelength range, then you can see that what that would mean is that we would only have the ability to distinguish 3 colours, because each individual wavelength of light would either stimulate one receptor or the other one or the third one. It's only because they overlap that we're able to distinguish a whole range of different hues, because what the brain is going to receive are separate signals from each of the 3 receptors, and any given wavelength of light could in principle be stimulating 2 or even all 3 receptors, and it's the relative strength of the signal that it's getting from each receptor which is interpreted in terms of the particular colour observed. So we can see how that works if we just sort of scan through, starting from the short wavelength end. So you can see just above 400 nanometers that we're getting strong stimulation of the short wavelength receptor and much less of the other 2. And that's what our brain is told, that we're getting big signal from one receptor, not much from the other 2, and it interprets that as that sort of violet colour that we get at that end of the spectrum. As we start moving on though towards sort of 450 nanometers and beyond, you can see that now we're getting a substantial signal from the medium range receptor as well as the short wavelength one, and also some from the long range receptor too, because the medium and long are only slightly displaced relative to one another. And you can see from the colours at the bottom that that's what our eye interprets as blue. So blue is what happens when you get a wavelength which is giving stimulation particularly of the short and medium range receptors, but less from the long range one. And if you think about the blue solution we looked at, remember that that wasn't just transmitting a single wavelength, it was transmitting the whole range of colours through the violet and blue and green and indeed yellow regions, and it was only really the red that was being taken out. And you can see that what that's going to mean is that you're going to get strong stimulation of the short wavelength receptor and of the middle wavelength receptor, but it's the long wavelength one particularly that's going to get less stimulation than it would do from white light. So as far as your eye's concerned, it's getting the same story as it would do from pure blue light, which is strong stimulation from the short and medium wavelength receptors and less from the red one. So we can't tell the difference. A pure blue single wavelength can look completely identical to that whole range of wavelengths just because the level of stimulation of the 3 receptors shows the same pattern. As we continue on past 450 and on towards 500 nanometers, you can see eventually the colour we perceive starts to change from blue into sort of turquoise and then into green, and what that correlates with is the signal from the medium range receptor now eventually becoming comparable to and finally overtaking the strength of the signal from the short wavelength receptor. So that move into green is dictated by the relative strength of the signal from those 2 receptors, particularly shifting in balance in favour of the green one. So when we had a green solution, remember that the light transmitted was primarily green with just a little bit of red and blue at both ends, and that's going to correspond to stimulation primarily of the middle receptor, with still some from the other 2 but less. And again, that's indistinguishable to the eye from for example a wavelength of around say 540 nanometers or something, where again you'd get strongest stimulation of the medium receptor and less of the other two. When we think about yellow, remember what we saw with the yellow solution was that what was missing from the spectrum was the blue light. So we were getting the full range from green through to red. And you can see that what that's going to correspond to is pretty much equal stimulation of the medium and long range receptors, but much less of the short range one. So yellow is what we see when the medium and long range receptors are getting comparable levels of stimulation. And of course that can also be achieved by pure yellow light. A single wavelength corresponding to yellow would also have the effect of stimulating those two receptors equally. So you can see how well this model works for explaining how a range of wavelengths can be indistinguishable to the eye from a specific wavelength. Once we move up beyond sort of 650 nanometers, you can see that the long wavelength receptor is still being stimulated, but the other two are getting very little stimulation, none at all really, for the short wavelength one by that point. And of course that's what we see as red, where the signal is dominated by the long range receptor and we're getting very little from the other two. And again, that's what we saw with the red solution, that we were really only getting red light coming through, so only that receptor would be stimulated. So that's the essence of how colour vision works. And you can see that it provides a very neat interpretation of this apparently complex idea that single wavelength colours can't easily be distinguished from colours resulting from a whole spread of wavelengths, which is what tends to happen when light passes through a solution. So now we're going to get you thinking again, this time trying to make use of the three receptor model that we've established for colour vision, and see if you can use it to figure out what's going on when someone is affected by colour blindness. So this is quite a common condition where the affected person just sees colour differently from most people, and in particular tends to have quite limited ability to distinguish between different shades of colour in part of the spectrum. There are actually lots of types of colour blindness, but the most common ones are usually lumped together and described as red-green colour blindness, where the person just has quite limited ability to tell the difference between shades of colour across that whole range, really from green to red at the longer wavelength end of the spectrum. Now these conditions arise as a result of mutations in the genes that encode the photoreceptor proteins. Okay, so these cone cells contain light sensitive proteins, and if these proteins are altered slightly as a result of mutations in the genes that encode them, it can affect their response to light. So what we want you to do is to pause the video in a second and just see if you can think of possible mechanisms for red-green colour blindness involving mutation of the genes encoding these photoreceptor proteins. There are several possible mechanisms resulting in red-green colour blindness. So just see if you can think of as many as you can. So the simplest explanation for red-green colour blindness is that one or other of the medium wavelength or long wavelength photoreceptors is simply inactivated as a result of the mutation in the gene that encodes it, so that in effect colour vision now depends on just 2 receptors rather than 3. So these 2 pictures show you how that's going to work now if you only have 2 receptors, and you can see that at the short wavelength end of the spectrum you still can discriminate different colours, because the shade of colour is going to depend on the strength of the signal coming from the short wavelength receptor compared to the strength of signal coming from the longer wavelength one, and that's going to be effective at discriminating between shades of blue and turquoise and so on. But once you get above about 530 nanometers, you can see that the short wavelength receptor essentially no longer detects light. And so above that wavelength, only one receptor is being stimulated. So any light in the range above that is going to just appear the same because it's just a signal arriving at one receptor. And you can see that because the medium and long wavelength receptors have relatively close together sensitivity profiles, that's going to produce similar consequences, which is just that there is a broad range of wavelengths at the longer end of the range where you just can't tell the difference between different shades of colour. So both of those conditions are actually known, although in fact the most common type of red-green colour blindness is one where both the medium and long range receptors are working, but the mutation results in their maxima being shifted much closer together, so the 2 curves mirror each other quite closely, and that makes it difficult then to distinguish between different colours, because you're going to be getting quite similar signals from both receptors over a broad range of wavelengths. So here's another one to think about. So here we've got 2 people, Zebulon and Mehetabel, both have red-green colour blindness, but one of them has a mutation where the long wavelength receptor has been knocked out, while the other has a condition where it's the other one, the medium range receptor, which has been knocked out. And your job is to see if you can think of how you could tell which one was which just by doing experiments in which they tell you how they respond to different stimuli that you might provide for them. So again, pause the video while you think about how you could come up with an experiment which would let you tell these 2 who has which condition. Well, probably the simplest way to do it is to look at what happens at the extreme long wavelength end of the spectrum, above about 650 nanometers. So in this range sort of here. And what you can see is that if the long wavelength receptor has been knocked out, then actually when you're depending on this receptor, you're going to have very, very little colour perception, or rather light perception at all, above about 650 nanometers. So essentially, when you have long wavelength red light, you're not going to be able to see very much at all if you have that condition. Whereas if you have this one where it's the medium range receptor which is knocked out, then you're still going to be able to see in this range because you still have some albeit lowish level, but that's always true with red light. You're still going to have the same as anyone else in terms of your ability to perceive red light at in this range. So that would be the simplest way to distinguish between the two, would just be to show them quite long wavelength red light and just ask how much they can see. Of course, there is the other side of that coin, which is that in this part of the spectrum, in the sort of blue region, you can see that the person who has the green receptor, the medium range receptor, knocked out, has quite low sensitivity in this part of the spectrum. So this minimum here, where the combined receptor sensitivity of both receptors is really quite low, and it's quite a lot lower than it would be in the case of this individual where the long wavelength receptor has been knocked out. So you get a sort of dimming in the middle of the spectrum if you have the medium range receptor knocked out, but it's not as easy to pick that up. This crossover point is actually also quite interesting in the sense that what that crossover point represents is when you're getting equal stimulation of both receptors. So of course you would see the same thing there if you had light at that specific wavelength where they cross. You would see the same thing as if you had pure white light, because white light has all of the colours, all of the wavelengths at equal or roughly equal intensity, and so as far as your two receptors were concerned, white light would give the same response as that specific wavelength. So that's something that you don't get if you have the three receptors. There is no single wavelength which looks the same as white normally, but there is in the case of these individuals, and you can see that the wavelength at which that occurs is different. So that would be another possible way to distinguish between the two, is to find the single wavelength of light which looks the same as white, and you can see that that would be at a longer wavelength for this individual where the medium range receptor has been knocked out than it would be for this one. It would be really interesting to be able to understand how these photoreceptors work at the molecular level, to understand how these proteins in the cone cells are able to interact with light, and how this interaction in turn is able to stimulate nerve impulses which are carried to the brain and subsequently interpreted as colour. So that's something we will try and return to later, but we're talking there about a very complicated photosensitive system, and before we can hope to make any sense of that, we need to understand in a much more straightforward system how light interacts with atoms and molecules. So we're going to start with this, which is something that's probably familiar to you from school, which is the idea of a flame test. So what you probably already know is that if you have a Bunsen burner flame with the air hole wide open, that flame has very little colour. It's just a very pale blue colour. But if you introduce a small amount of a metal compound into the flame, you can get these strong characteristic colours. And what we're going to do is think about how that works. And we're going to focus in particular on the bright yellow one that you can see in the middle there, which is the flame obtained when sodium compounds are put into a flame. So the way we're going to approach this is once again by doing an experiment with the diffraction to separate out the spectrum and just have a look at what that yellow light emitted by a sodium flame actually comprises. So here's our experimental setup. Once again, the optical system inside the big box. There's the CD to act as the diffraction grating, and there's a pinhole above it, as well as the camera trained on the CD. And here on the outside we have a camping stove, and that is going to act as to provide our flame source. So here's the flame actually lit, and what I'm chucking into it with the spoon is some sodium bicarbonate, and that's what's causing the intense yellow colour of the flame. So let's have a look inside and see what its spectrum looks like. Okay, so now we're looking at the feed from the webcam, and first we're going to have a look at what happens if we just shine the white light from the torch through the pinhole, just to give us a sort of reference spectrum. And there it is, so you can see that we see the full range of colours from blue through to red, as we expect. So now if we take the torch away, we should be able to see what happens if we then replace that with the flame. So we take the torch away, and there you can see the flame. So it's a bit flickery because it's not as bright as the torch, and obviously flames flicker, but you can see, I think, quite clearly that what we're getting that's causing that yellow light is just a very narrow part of the spectrum in the sort of yellow to orange region that's showing up, causing that colour. And that's the interesting thing then, that the colour that causes the yellow flame, although it looks kind of similar to the yellow colour that we saw from the yellow solution, the origin of it is completely different, because where in the yellow solution we were getting transmitted light spanning the whole range of the spectrum from green through to red, with this we're just seeing a very narrow, well-defined region of yellow light. Okay, so here is a summary of that then, comparing the broad range of wavelengths that add together to give you white light with that very narrow defined region of the spectrum which corresponds to the yellow light emitted by the sodium flame. So what we're going to get you to do now is to think about what the significance of that result is. So basically what we want you to do is to see if you can explain what happens when a metal compound is placed into a flame. So we'll just tell you that the first thing that happens is that a little bit of the compound at any one time will tend to break down in the heat of the flame to form its constituent elements. So you can assume that there is a small amount of elemental sodium, in other words of sodium atoms, present in the flame, and it's those which actually give rise to the colour. So what we want you to think about is what's going on to cause the colour to be emitted, and in particular what's the significance of the fact that it is a very specific colour corresponding to just a very narrow region of the spectrum, rather than white light, which would correspond to a whole range of wavelengths across the whole spectrum. So have a think about that, pause the video while you see if you can work out what that story is, and then you can move on and see what the conventional wisdom is. Okay, so we can understand this in terms of energies. So the idea is that an atom can have various different energy levels, but that under normal conditions it will tend to exist in the lowest energy state that is accessible to it, which we call the ground state. Okay, so this represents an atom sitting in its ground state. But of course in the heat of a flame, the atom is able to pick up energy. And if it picks up sufficient energy, it can move up into a higher energy state, which we call an excited state. So here's an atom which has now picked up some heat energy from the flame and is sitting in an excited state. However, it's not going to be stable in that state, because atoms will always tend to drop back down into their lowest energy state after really quite a short time. But of course, in order to do that, they have to lose energy in some way, and the way they're going to lose that energy is in the form of light. So that's what happens next, that as the atom reverts to its lowest energy state, it emits a photon of light. And that's the origin of the emission that we see in the flame. And in the case of a sodium atom, that light is dominated by this yellow colour. And that comes from this transition where the atom drops from its lowest energy excited state back down to the ground state. Of course, there are other possible transitions because there are other excited states up there, but the other transitions either occur much more rarely and therefore don't give us bright light, so we didn't detect them in the spectrum, or in other cases occur at too short a wavelength, so that if they're in the ultraviolet we wouldn't see them. So actually that yellow colour really comes just from this one transition in the case of sodium. So that's a story which you may have heard before, and which certainly would probably seem reasonable based on ideas that you've had about energy levels in atoms in the past. Now, to be really clear about the significance of the fact that emission from that sodium atom occurred just with that single very specific colour, we need to use this equation, which is called the Planck-Einstein equation. And what it relates is the frequency of the emitted light to energy. Now, frequency, remember, is related to the wavelength. So a specific colour corresponds to a specific wavelength, but therefore also to a single specific frequency. So what this equation tells us is that the yellow light emitted had a specific frequency, or at least a very narrow band of frequencies. And that, multiplied just by Planck's constant, gives us an energy. And that's the energy of a single packet of light. So light can be thought of as being composed of energy packets, which we call photons. And what this is saying is that the specific colour of light corresponds to a specific frequency, and therefore to a specific energy of the emitted packets of light. Now, what's the significance of that? Well, the significance is that when the atom jumps down from an excited state to the ground state, it gives out its excess energy all in one go. So the energy is released not in sort of dribbles, but in one single packet. And the fact that those energy packets all have the same energy tells us that the excited state has a single very specific energy. So if the atom could have all sorts of different energies, then you'd get all sorts of different photon energies being emitted as it jumped back down to the ground state, and therefore you'd get a whole range of frequencies corresponding to, if it was the full range, white light. And the fact that we don't see that is therefore, as we said, evidence for very specific energy levels in atoms. Now, that may well not shock you, because you've probably been exposed at school to diagrams of energy levels in atoms. And, you know, you're just sort of told, well, this is the way it is, so you kind of accept that. But I think it's really important at this point to stop and think that that's actually really weird. So let's think for the moment of an analogy of an atom, and just think about a tennis ball that you're holding in your hand. Now, you can throw that tennis ball up in the air, and if you throw it gently, it might reach a height of 10 centimeters and then fall back down. If you throw it a bit harder, it might reach 15 centimeters or 50 centimeters or 2 meters or whatever. You know, you can give it any amount of energy you like, and therefore it can go up to any height you like, limited only by really the strength of your arm. But what we're saying is that if it was like an atom, it wouldn't be like that. If it was like an atom, it would seem that you could throw it up to 10 centimeters, and you could maybe throw it up to 15 centimeters, but you couldn't throw it anywhere in between. It would be impossible to reach a height of, say, 4 centimeters or 12 centimeters, because there are only these defined excited state energy levels accessible. And that just seems incredibly weird, because obviously that's nonsense in the case of a tennis ball. And so we need to ask ourselves, well, why is it that atoms can only have these specific energies? It's extremely bizarre if you think about it. And when this idea first arose in physics, it was hugely controversial. And people took an awful lot of persuading that it could be true, because it just doesn't seem to make any sense. And yet the weight of evidence that we have now says it is absolutely true, and this is the correct interpretation of what's going on. And it's the foundation of what's called the quantum theory. And that's where we're going to go next time. So we're going to take this idea of specific energy levels in atoms and molecules and explore that in much more detail. And then once we've made some sense of that, we'll come back and see how it relates to the interactions of molecules and light. But that's it. That's the end of today's session.